Hello everyone, my name is Priyanka. My qualification is MSc BA and CSIR NET. Today I am going to revise some questions based on the topic Mendel's laws of inheritance and monohybrid, dihybrid and trihybrid crosses. So let's go to our first question, that is Mendel's laws of segregation as applied to the behavior of chromosomes in meiosis means that what was the Mendel's laws of segregation. In Mendel's first law, which was Mendel's laws of segregation, he tried to explain about purity of gamete. When gametes will form after meiosis, they will have only one kind of allele. You know that alleles are various form of genes. One gene can have two or more than two alleles. So, at the time of meiosis, when homologous chromosome will separate from at the time of metaphase 1, they will go in two cells and they will have separate alleles. I am just trying to say that this is the metaphase first condition when the two homologous chromosome, you must all have known that P has 14 chromosome or 7 pair of chromosome. Why pair? Because two chromosomes are homologous. One is coming from male plant, one is coming from female plant. And what is the meaning of homologous chromosome? Because they will have alleles for same gene. Here if you have capital T, here you can have small t. They have allele for same gene. That's why they are called homologous chromosome. When they will divide at metaphase, one homologous chromosome will go towards one pole with this spindle apparatus. This is spindle apparatus which attach itself with the centromere of chromosome, and the other homologous chromosome will go to the other cell with the spindle apparatus, and they will go opposite poles and in opposite cells. They will go to two cells. That's what they are asking here, or they want to understand that what is the Mendel's laws of segregation. It actually means that gametes are pure. They will have only one kind of allele at one time. For example, when this kind of plant which is heterozygous for length tt will form gamete, the gamete will either have T in it or have small t, recessive T in it. They cannot have both the alleles at one time. Why? Because at time of meiosis they will go to the separate cells. That's what they are asking here. Alleles of a gene separate from each other when homologs separate in meiosis first. This is the statement I was trying to, this is the point I was trying to explain it here with the homologous chromosome. Genes on the same chromosome will show 50% recombination. In the Mendel's law, there were no explanation about recombination. Alleles of a gene will be linked and passed on together through meiosis. Concept of linkage were not understood at the time of Mendel. In fact, if Mendel know about linkage, then his theory of inheritance would be little bit different from now. At that time he was not aware of the linkage. So our answer should be B. Let's check. Our answer is B, because a gamete may carry either the dominant or the recessive allele, but not both. You can find it, you can understand it with the Mendel's experiment. In his first experiment, he found only dominant character in first generation, but in second generation, he found both dominant and recessive characters into 3:1 ratio. But 3:1 ratio explains that the recessive character was present in F1 plant, but it was hiding. It was not expressing itself. But when gamete form for F2 generation, the recessive gametes separate from the dominant gamete and form new combination in F2 generation, that is 3:1. That's what they are asking. Our second question is, the phenomenon of independent assortment refers to. What was the phenomenon of independent assortment? If you don't know, you check my channel. I have made videos on the Mendel's laws of inheritance and crosses. First option is expression at the same stage of development. No, phenomenon of independent assortment did not explain it. Unlinked transmission of genes in crosses resulting from being located on different chromosomes. When Mendel conducted dihybrid cross, he took two characters at one time, but these two characters were not located on same chromosome. They were located on different chromosomes. That's why they transmitted independently of each other. One character was not affecting transmission of other character. How? Let me explain you. In Mendel's dihybrid cross, he took two characters at one time, and you know that P has 7 pair of chromosomes. So it means that when he took two characters, those characters must be present on two different chromosomes. For example, he took seed color yellow and green. Yellow and green. Yellow must be present on this, and the green allele must be present on the homologous chromosome of this. And he took other character that is seed shape, round and wrinkled. So round seed shape must be present on other chromosome, and wrinkled shape must be present on this homologous pair of it. So when these two characters will go to the next generation, they will go independently of each other because they are present on different chromosome, other chromosome. If both the alleles, that is yellow and round or green and wrinkled, would have been on same chromosome, so they may not go to independently of each other. They may have been linked. You all of you must know about linkage. When the alleles are present very close to each other on single chromosome, they may be linked and they can go from one generation to other in linkage. That's what the concept of linkage. But when Mendel did his experiment, all the characters he took in his experiment did not show any linkage. That's why they independently go from one generation to other generation. It means that this Y can go with either round shape or wrinkled shape, and this green can also go with this round shape and this wrinkled shape. So they will form new combination. For example, they can form yellow and round, yellow and green, round and green, and round and yellow, and wrinkled and green, and wrinkled and yellow. They will form new combination. When he took parent varieties was only two, yellow and round and green and wrinkled. But when he obtained F2 generation, there were two new combination that was not present in parent, that was yellow wrinkled and green round. These were new two combination. Why new combination happens? Because of unlinked transmission of genes which are present on different chromosomes. So this must be the B must be the answer. Third option is association of an RNA and a protein. There is no association of RNA and protein in Mendel's laws of inheritance. Independent location of genes from each other in an interphase cell. Mendel's laws of inheritance do not tell about location of genes in interphase cell. So the answer is B. Let's get to the next question. One of Mendel's pure strain. What is pure strain? Pure strains means that homozygous condition. Pure strains of pea plant had green pea, and you know that green color of the pea plant is recessive character. It means when it is homozygous recessive, it means that it must be like this. Pure means homozygous. So when it will form egg or pollen, it will have only one kind of egg or pollen, that is this. How many different kinds of egg could such a plant produce with regard to pea color? It can produce only one type. 1, 2, 3, 4. The answer must be 1. List all 3 gametes produced by the following individuals. AaBbCc. Here you can see Aa and Bb are homozygous, but only C is heterozygous. It means that it will produce 2 types of gametes. When gametes will form, the chromosome number will reduce to half because of meiosis. So we will write it like this. It will form 2 types of gametes, A, B, C, A, B and small c. This is capital C and this is small c. So this will form 2 types of gametes. And other individual is small a, BB and CC. It will form, see how many kind of gametes. Let's try to understand how we will form gamete when you will have this type of situation. See, you have to write it like this. A. Since this is homozygous, you will write only one A. Then you will multiply it with both the alleles of B, that is capital B and small b. Then you will multiply capital B with both the alleles of C, that is capital C and small c. And you will also multiply this small b with capital C, small c. Then you can write, you can know how many gametes will form. See, you can see here, write it like this: capital small a, capital B, capital C. For this you will write small a, capital B, small c from this A, B and C. For this small b you will write small a, small b, capital C. For this you will write small a, small b, small c. You will form 4 kinds of gametes with this combination and 2 types of gametes with this combination. You should always learn this method, this very method to form gametes. It's very easy and you can form easily in exams also. So let's check our option. The first option is 2, 4. I think it's right. First option is right. Let's second option. In second option, this is small b and small b is not present in 1, so it must be wrong. In third option, both the gametes are AaBbCc. That is not right because there is a small c in first option. So third option is also not correct. In fourth option, here in first combination ABC is right, but small b is not present in first option, so it's not also correct. So first one would be answer. Answer is first. If a diploid cell contains 6 chromosomes, how many possible random arrangements of homologs could occur during metaphase 1? See, I have made a video on mitosis and meiosis, and there I have clearly explained the difference on metaphase 1 of mitosis and meiosis because it's very important. In meiosis, recombination and crossing it over make the gametes different from each other, but the combination at the metaphase plate, how homologs arrange themselves at the metaphase plate, also add the variability in the gametes. It also increase the variability because at metaphase first of meiosis, chromosomes can be arranged in any manner. How? I will show you. In the question they are asking about 6 chromosome, it means 3 pairs of chromosome. So you can form like this. I am forming 3 pairs of homologs or 6 chromosomes. I am forming it with little bit similar color because they are homologous. Homologous means they will have alleles for one gene, same gene. So if one cell has 6 chromosome or 3 homologous chromosome at metaphase first of meiosis, how will they arrange? When at metaphase first of meiosis, these homologous chromosomes goes to different cells, two different cells. How the spindle will attach itself with the centromere of one homologous chromosome. So this homologous chromosome will go towards this direction and this homologous chromosome will go to this direction in two different cells. It means that homologs are separating from each other at metaphase first. So what will happen in one combination? These 3 will go in one cell and these 3 will go in other cell. But it can also change its position. Homologs can also change its position in a different scenario. This pink one can come here and this red one can go here. So the combination will change because this homologous chromosome and these are not same. They are similar but they are not same. This may have the dominant allele and this may have recessive allele. So when it will go here and this pink can come here, the combination is changed. They will have same gene but alleles are different, so combination will change. And in next scenario, this green can come here and this green can change its position to here also. They are free to change. Homologous chromosomes are free to change their position at the metaphase first, so they can have more varieties. And this blue can come here and this blue can go there. So it means that originally we one situation, then we can have 3 more situations where these 3 chromosomes can arrange themselves. It means that we have 4 types of arrangement at the metaphase first of meiosis. 3 pair of chromosomes can give 4 types of arrangement. it will increase the variability in gametes. That's why no two individuals of human being are same on earth. There are 6 billion people on earth, but no two individuals are like one another except twins, because of meiosis. And metaphase first arrangement also increases the chances of variability in individuals. It's a very important concept, you should always remember, and you can clear it more when you will understand meiosis and mitosis separately and difference between them. Next question is, determine the frequency of only one genotype BB capital L small l in the offspring of dihybrid parents BBL. It means that the parent is this, so it will cross with the other parent which is also the same, so it will form 4 type of gamete: B, L, B, L, B, L and BL, and this will also form the same 4 type of gametes. It means that on checkerboard you will keep this type 4 gametes here, this 4 here, this 4 here, and the same with here. When you fill this checkerboard, you will find that this combination where one is BB and other is L and small l will you will find this type of genotype only in two plants or offspring. Where I am showing you, you will find it here. You will find both the dominant. Here you will find this. Here you are finding this genotype, and other one is here. You will find only 2 individuals of this genotype, capital B, one capital L and one small l, 2 individuals. You can form this checkerboard and you can check yourself. See, so the offspring of dihybrid parents, you will find that, so you know that in the dihybrid cross you have 16 type of genotypes, 16 type of individuals in 16 type of genotypes. So from 16 you will find 2. It means that from 1 is 8, the ratio is 1 is 8. From 16 individuals, 2 are this type of genotype, so from answer should be 1/8. Answer is B. If an individual of genotype AaBbCcDd is test crossed, how many different phenotypes can appear in the progeny? This is tetrahybrid cross. You know that in the trihybrid cross, what is, first you should know what is test cross. Test cross means that when any heterozygous individual is crossing with the recessive parent, It means that this AaBbCcDd has been crossed with the recessive parent, that is AaBbCc and Dt. Recessive, all the recessive. What will happen? This will form 16 type of gametes. This tetrahybrid plant will form 16 type of gametes, and this will form only one type of gametes. So how many different phenotypes will appear? You can see yourself. This number will be 16. Answer will be 16. How? You can see how many gametes will form with this. You can form with this. Like this. Like this. D, 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 D. You have to multiply all the alleles with every allele. D, 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 D. You will find 16 type of gametes. When you will multiply 16 with one type of gamete, that is, this will form only one type of gamete, that is AaBbCd. You will form 16 combinations, 16 type of phenotypes. So you can check yourself. You can do this cross and copy and check yourself. I told you how to form gametes. So the answer would be 16, 8, 32, 64. The answer would be 16. If individuals of genotype AaBbCc are intercrossed, how many different phenotypes can appear in their offspring? You should know that when the numbers of gene pair are n, then the number of phenotype will be 2n and the number of genotype will be 3n. So here the number of gene pairs are 3. So the number of phenotypes will be number of phenotypes, and this is genotype. This is a rule, will be 2 upon 3, and the number of genotypes will be 3 upon 3. So here they are asking about phenotypes. It means that 2 multiplied by 2 multiplied by 2 means 8. The number of phenotypes in this trihybrid cross will be 8. Answer is C, 8. You should always remember that rule. If number of pair is n, number of phenotype is 2n, and number of genotype is 3n. You can do it for any number of pair of genes and you will find result. Next question is same. If individuals of genotype AaBbCc are intercrossed, how many different genotypes can occur in their progeny? Genotypes will be, if the number of pair will be n, the phenotype is 2n and genotype is 3n. The genotype is 3 to 3 because number of gene pair are 3 here. So 3 into 3 into 3 is equal to 27. So answer should be 27. Answer is A, 27. Next question is, a trihybrid cross is made between two yeast both with genotype trihybrid. This is trihybrid cross. I made a video on trihybrid cross and their genotypic ratio and phenotypic ratio. What proportion of the offspring will be genotype? Genotype is here, they are showing Here they are showing only one kind of genotype, that is recessive, and you know that in dihybrid cross or trihybrid cross or any cross, the recessive, only recessive individual is only one, in dihybrid also, in trihybrid also. And in trihybrid called total offspring were 64. So the answer should be 1 upon 64. The answer should be C. A man is heterozygous BB for one autosomal gene and he carries a recessive X-linked allele D. What proportion of his sperm will be BD? For human being, there are two. We write 2A. Why? The autosomes, all the autosomes are in 2. We have 46 chromosomes, but they are in 23 pairs. So we write it like 2A, 2 autosomes, plus for men X and Y. The men have 2 pair of autosomes and one X chromosome and one Y chromosome. But here they are saying that one autosome is have 2 types of allele. It means different. Either it will be 2AB capital B or 2A small b. So it will be like this: 2A capital B or 2A small b. 2 types of autosomes in this situation. And X is also have a linked gene, that is D. It means that it will also arise 2 situation: X, D or Y. This situation will also have 2 types of multiply: X, D or Y. It means this person that have one autosome is heterozygous and one X have D recessive linked allele, it will produce 4 types of sperm. Because this is man, it will produce sperm, 4 types of sperm. In one sperm it will have 2A capital B and X linked with D, because you know in gametes only one pair of chromosome can go. So it will have 2A 1A, and in other it will have AaBbY in one sperm. In other scenario it can have A with small b and X with D, and in other scenario it can have A with small b and Y. So it can form 4 types of sperm because of this condition and this condition. I hope you have understand it. So the option is 1 upon 64, 1 upon 4, 1 upon 16, 1 upon 2. The answer will be C, 1 upon 4. 4 types. Thanks for watching. This was the today's. So subscribe my channel and watch my video and do comment.